Okay, thanks everybody for coming. This is PhD Defense by Jeremy Rose. That's the title. I'll let him go over it. Jeremy came to us from Santa Barbara, which was basically his second bachelor's degree. He first went to the University of Washington and got a degree in electrical engineering. And then he decided to do some work in the medical field, and so he started on a master's, I think, in medical engineering or something to that effect. And did a bunch of stuff, invented a few things and so forth, but he finally saw the light and realized that, working inside in a air, dry sort of environment with a bunch of human-related things is not really what he wanted to do. And so he went to Santa Barbara, where they had a program for old guys who had already gotten a bachelor's degree in something else, and did a biology degree. And there he went with people who were doing field stuff, and he got excited about that. And when he was done, he applied to a number of graduate schools, and luckily we were able to get him to come here. Jeremy has had the pleasure of having a 3-year NSF pre-doctoral proposal for support of part of his time here, and also worked with us on the Omegas project, which was an NSF-funded project focused on ocean acidification, which is going to be the focus of his talk today. So, I looked for tried to get some embarrassing pictures of him, but I didn't talk to the right people.
He clearly didn't try very hard because it wasn't taking much effort.
Yeah. It also looks like everything is going to be all right. So,
which I happen to have that shown.
There's one thing. They're all over the place. So,
Thanks, Bruce. Thanks for that intro. So the topic of my dissertation has really been focusing on the ecology of ocean acidification. And in particular, it's integrating the effects of natural pH variation on biogeography and organismal performance in rocky intertidal communities. So we're looking at rocky intertidal systems and how they respond to natural variations in pH that might be associated with ocean acidification. Here's an outline for the talk for today. I'll give you some basic background on the chemistry of ocean acidification and how we see our changing carbon climate. I'll talk a bit about the diversity of organismal responses to OA that is in this ever-expanding literature knowledge base that we have. And then I'll talk also about parallel efforts to understand pH variability in the oceanographic climate system. And then I'll introduce my PhD research, talk a bit about calcifier distributions, and move on to 2 questions that are fairly similar but deal with very different species. And that's looking at the impacts of natural pH variation on purple sea urchins and also on California mussels. And then I'll close with some concluding thoughts. So first, the basics in our changing atmosphere driving our changing oceans. If we start by looking at the red line, this is a line that many folks who have looked at changes in atmospheric carbon dioxide are pretty familiar with. It's the classic Keelan curve that measures carbon dioxide concentrations in the atmosphere since about 1957, all the way through continuous measurements today. And you can see that there's this generalized, fairly linear decline in atmospheric carbon dioxide over time. About 1990, a marine station was set up, Station Aloha, to take a look at the effects on the oceans as well. And what we can see is that the carbon dioxide for the oceans, as shown by the blue lines, is increasing at roughly the same linear rate as that for the atmosphere. So these two appear to be very linked. These changes in carbon dioxide for the oceans lead to changes in pH in the oceans as well. So rising carbon dioxide is driving a decline in pH over time. And in just this 20-year period, you can see a decline of about 0.05 units. The way this chemistry works is relatively straightforward. As we build up the pressure of atmospheric carbon dioxide, it forms a pressure gradient at the surface of the oceans. That pressure gradient then forces carbon dioxide into the ocean waters, where it reacts very quickly with water molecules to form carbonic acid. And carbonic acid quickly dissociates into hydrogen ions and bicarbonate ions, as we see all the way over here on the right-hand side. And now if the chemistry stopped right there, the pH of the oceans would be quite a bit lower than they are today, because it's this accumulation of hydrogen ions that drives the pH lower. Okay? But the oceans have this buffering capacity through the availability of carbonate chemistry that can, in a sense, take some of these hydrogen ions and bind with available carbonate ions to form additional bicarbonate, and therefore help keep the effects of this pH change fairly mediated. The outcome is that it removes carbonate ions from the water column, and these carbonate ions may be very important for a wide variety of organisms that biomineralize calcium carbonate structures. They have body parts made of calcium carbonate. So this reduction in carbonate here to help address this change in pH may have an effect on these calcifier organisms, and has drawn a lot of attention on calcifiers and the effects of ocean acidification. I could have had a million pictures up here showing a wide diversity of organisms that have calcified components, but this gives you just a general idea. It includes planktonic species such as forams, coccolithophores, tiny little pteropods. We have mollusk representatives with their very strong hard shells here with snails and bivalves, echinoderms, including urchins and sea stars. We even have worms that make casings out of calcium carbonate, and even some algae that rely on calcium carbonate for structural support. So a huge, broad diversity of marine calcifiers could be potentially affected by pH change. One of the biological influences that can help mediate these responses, or can dictate some of the responses for these organisms, has to do with the primary form of calcium carbonate. So not all calcium carbonate is the same. The way the crystalline structures lay down upon each other in three-dimensional shape leads to two main types. Calcite shown here, and aragonite shown here. And this image on the right-hand side actually shows a shell that contains both aragonite and calcite in two separate layers, with aragonite on the bottom, calcite on the top. So you can see they're quite different structurally. It turns out that there's another difference with calcite, in that calcite can also have varying degrees of magnesium that are incorporated into the crystalline structure. So we can further divide calcite into low magnesium calcite, and as you see on the far right, high magnesium calcite as well. And why I divide these three is that it turns out that at a set pH, set conditions, they have different solubilities. So high magnesium calcite is more likely to dissolve at a set pH than aragonite, which is more likely to dissolve than low magnesium calcite. Okay? So depending on what type of minerals these organisms use, they could be subject to increasing solubility stress. It's important to recognize that these solubilities for all three decrease with decreasing temperature, so there's very much a temperature effect on the solubility as well. And that comes into play when we start to look at large-scale changes. So now we're looking at aragonite saturation, that middle type of calcium carbonate. And we're looking at the values of saturation state, which values of about 1 are considered the lowest threshold at which organisms may be able to build up calcium carbonate. Below 1 is complete dissolution at that point. And the first thing that jumps out is if we look at just one image, let's take the 1875 image, for example, you can see that there are broad latitudinal trends in the aragonite saturation state. We have much higher saturation states, as indicated by the cooler colors, around our equatorial region, and lower saturation states towards the polar latitudes. And that pattern repeats regardless of what year it is that we're looking at. What you can see, though, from this historical reconstruction is that conditions were generally better than they were in 1995. Saturation states are getting increasingly lower. And as we take those forecasts forward into the coming century, we can see that it gets even more dramatic towards lower calcium carbonate, towards lower aragonite saturation states. So we have this encroachment both decadally and then due to temperature effects from latitude towards the equatorial region of lower aragonite saturation state. And this rate of change that we're seeing is unprecedented in the geologic record. It's the speed at which really just hasn't been seen in at least the past 25 million years of the records that we have in hand. So with that basic chemistry in mind, we can talk about how organisms might respond to ocean acidification. And this is just one study, and I don't expect you to be able to read all of the details on this figure on the right-hand side, but I want you to get a general sense of those figures. So I've set it up to show that each x-axis in each of these figures corresponds to aragonite saturation, with low aragonite saturation towards where the two axes meet, and enhanced aragonite saturation on the higher end of the axis. And then we have net calcification on any one of these y-axes as well. The species that I'm showing here on the left-hand side correspond to the figures on the right-hand side. So the top three or the top three, etc. And the main thing I want you to draw out of this is that there's a diversity in patterns into how these organisms respond to changes in aragonite saturation. For example, we have 6 organisms down here that have higher calcification at higher aragonite saturation states and lower calcification at lower aragonite saturation states. So this would agree with the main hypothesis of the chemistry driving these declines in calcification ability. We have a few organisms that are in the middle that have more of a sense of a threshold value, where conditions are relatively, responses are relatively constant and flat. And then as we dip down in saturation state, you get this drop off in performance. Looking higher up, you can see that we have some species exhibiting a unimodal curve, where there appears to be an optimal aragonite saturation state with reduced performance on either side. And then all the way at the top, we have the converse of what is going on here, where those performers are actually seeing greater calcification at lower aragonite saturation states than they experience at higher aragonite saturation states. So, kind of spreads the gamut. This was one study that looked at all these organisms. And there have been a huge exponential growth in studies that are showing that really variety is just extreme from one species to the next in their responses to changes in aragonite, PCO2, and pH. So that's one of the influences, but we talked about the one influence of calcium carbonate and the type of calcium carbonate being a biological effect. Another biological effect would be a developmental stage. So many marine organisms, they release their eggs and sperm into the water column where fertilization can occur. And then as fertilization occurs in the water column, the larvae then develop as pelagic larvae out in the water column, sometimes for days, sometimes for weeks, months, before they then settle back into the benthic habitat and metamorphose into adult form. And a meta-analysis that has looked at studies across the effects of different adults or different life stages have shown that pelagic larvae are generally more sensitive to reduced pH than juveniles or adults. So it appears that the developmental stage can play an important role in responsive organisms as well. Third way in which there could be biological influences include treatment-mediated sensitivities. I have 2 examples here for this. And one is another meta-analysis that has looked at responses across a variety of OA experiments and found that taxa that have higher proportions of calcified components exhibit increased sensitivity. So, for example, your heavily calcified mollusks are going to be more sensitive than your crustaceans, which are not as heavily calcified. Okay. And activity level can also play a role. So, more mobile species tend to have more exercise-induced changes in their metabolic systems, and therefore have better regulatory control of their extracellular pH, and are less dependent on the pH of the environment they're around. So mobility can play a role in the way these organisms have adapted to manage their own internal pH environment. Turns out the environment can also influence how organisms respond to ocean acidification. One key area is resource availability. And I highlight a couple examples here in which Olympia oyster larvae were shown to respond additively to the cross effects of food and pH. And what I mean by that is if you reduce pH, the larvae suffered. If you reduce the food, they suffered. If you reduce both the food and the pH, then they suffer the combined effects of both. So there is an effect of food availability shown for these Olympia oyster larvae. Blue mussel juvenile growth and calcification have been shown to depend chiefly on food supply and not pH. So experiments that have changed the quantity of food available along with changes in pH have found that really food is the main driver for these juvenile growth among the blue mussels. Temperature can also play a factor. Again, with blue mussels, we see that shell strength is adversely impacted by warming, but not reduced pH. So their sensitivity, again, to another environmental parameter that's not necessarily a pH parameter as well. And then we can see that the large purple sea urchin metabolism was additively reduced by both increased temperature and reduced pH. So here again, we have two effects working independently that when combined add up on top of each other for an increase in negative effects of those two. Timing of low pH events can also be important. The study is really, it's pretty cool the way in which they approached it. They established mean conditions just above 8.0, which fits the global mean today. And they ran two experiments, both of which had a mean of about 8.01. One experiment kept it constant. One treatment kept it constant. And the other treatment varied in a stepwise fashion around that mean. So that the mean value was the same, but there was variation around that. And the study by Cornwall and colleagues showed that there was an additive effect on coral and algal growth because of the stepwise pH variation around the mean. So it's not just a matter of the mean that matters, but how much variation there is in that. So we've gone through, there's biological mechanisms that can certainly affect the way organisms respond to pH changes. And there's a variety of environmental mechanisms as well. And as this knowledge base has developed on understanding biological effects, we've also seen parallel efforts to better understand natural pH variability. Turns out one of those systems that's received some attention is our system off the coastline here, California Current Large Marine Ecosystem. It extends from roughly Alaska down through Baja California along our coastline. And it operates under a general process of upwelling. The way upwelling works is a seasonal process. It starts around April and continues with varying degrees of intensity through fall. The process works by winds coming down from the north. Those northerly winds coming down, it pushes, because the spin of the globe tends to push surface water out to sea. And as that surface water gets pushed out to sea, deeper water then upwells and fills in the gaps that's left by that surface water due to the blowing of the wind. So the effect of these upwell waters coming in can change the environment, in the nearshore environment, in several ways. Often those waters are colder that are coming in. They tend to be higher in nutrients because this decayed matter has sunk. It's not available to be brought in towards shore. That can increase primary productivity. We can see changes in salinity, in mixed layer depth, a variety of environmental factors that are associated with this upwelling driver of processes along the California current system. Turns out, upwelling can also lead to changes in pH conditions along the coastline as well. So what I'm showing here on the right-hand side is a series of field cruises that were conducted by a group out of NOAA, led by Richard Feely. And in 2007, they took a series of transects. You can see the numbers and the lines leading in towards shore, despite like a coffee addicted hand. So you can see those lines running in towards shore. And what they were interested in doing was understanding the pH environment on a larger scale and how variable that was across the California current system. So this graph is showing the depth at which low pH waters were found. Low pH waters in this case were pH about 7.75. And you can see that they found low pH waters all the way up towards the surface, right here, just off the coast of southern Oregon and northern California. Now I want to point out this is not a single snapshot. You have to recognize that each of these points was taken at a different time, so it's a composite over the course of the season, which indicates there might be quite a bit more variability than what's going on right here. It's not that always this is going to be the highest that pH reaches towards the surface. It could be reached closer towards the surface in other locations as well as upwelling conditions change. But this was the first really strong evidence of this mosaic of pH along the coastline. As Bruce mentioned, my work has been a part of a larger consortium called OMEGA, the Ocean Margin Ecosystems Group for Acidification Studies. And that group has deployed a series of pH sensors along the coastline over the last few years. And this is, Francis provided me with this figure that summarizes some of the pH data that's coming back from this system. So what we're showing in this figure is latitude. So we've got pretty much the whole latitude from Southern California up into Oregon over the course of time, May, June, July, with the hotter colors representing lower pHs, the colder colors representing higher pHs. And what we can draw from this is it's really dynamic, right? We look just at the north, you can see there's periods of very high pH, low pH, high pH, low pH, high, low for a long time, and very low. So in space, or in time, it's very dynamic in this particular location. If we look and compare space, we can see it's also very dynamic in terms of space. So in this middle portion where we have very low pH here, we get some excursions of low pH towards the south. But here, very low pH towards the south and high towards the north. So it really does highlight that this is a mosaic pattern. We don't have a strong latitudinal pattern in pH, but it varies depending on where you are along the shoreline and what physical processes are at play. So all of these observations are really what feed into my research. And the main motivations for my research are really fourfold. First of all, it's the recognition that pH is a calcifier stress. So we have a lot of OA lab studies that do indicate broadly negative effects for calcifying organisms. We've also seen, though, that there's diverse influences on these pH responses, whether it be biological or environmental. So with this diversity in mind, we can start to think about scaling up to ecological complexity, where the environment is changing in multiple ways. And we can address how some of these diverse influences interact with pH on organisms in natural environments. And it turns out that we have this excellent national laboratory right off our coastline that presents a mosaic of pH variability, where we can now take a look at how organisms are responding in natural environments to ocean acidification stress, or natural variation in pH along the coastline. My overarching research question is natural pH variation relevant to the performance of rocky intertidal calcifiers? And that led to 3 follow-up questions that essentially drive the chapters of my thesis. The first is, how are calcifier communities distributed along the California current large marine ecosystem? Are calcifiers evenly distributed, suggesting that perhaps there's no pH effect, or do we see some sort of pattern that's out there? Second is, how is the performance of key rocky intertidal calcifier, the purple sea urchin, influenced by natural variations in pH, among other factors that are changing in the environment? And a third question is since we don't assume that sea urchins are the model of the entire system, we want to take a look at the role of another important calcifier, and this would be the California mussel, to see how pH and other environmental factors can influence this California mussel as well. So we'll start out here with how calcifier communities are distributed along the California current large marine ecosystem. General methods that we applied, and I had a long list of co-authors on this, which I'll talk about with why in just a moment here. But essentially what we did was we took PSCO biodiversity surveys. PSCO is the Partnership for the Interdisciplinary Study of Coastal Oceans. And they had this ambitious, incredible survey series from 2000 to 2003 that looked at 49 different rocky intertidal sites annually, spanning 16 degrees of latitude on the U.S. West Coast. So I'm taking the Feely pH image and I'm giving you a sense of the range of sites that were covered over the course of those surveys. So incredibly detailed surveys in which diversity and abundance were assessed at 3 different time heights. And every organism that was encountered was identified to as low a taxonomic level as possible out in the field. The bulk of the effort for this work really came from myself and many other folks in the Mengi lab who went through the effort of taking this list of species and then classifying whether they were non-calcifiers or calcifiers. That's very easy. The next tricky step, though, was what type of calcifier these organisms were. And that involved a very detailed literature review to go through and identify the primary calcium carbonate forms, either the high magnesium calcite, aragonite, or or low magnesium calcite. And we did that because we wanted to get a sense of perhaps there's some sensitivity to these systems based on the simple chemical dynamics at work on these forms of calcium carbonate. So first of all, who are the calcifiers in the rocky intertidal systems? We broke it up into two different groups, mobile taxa and sessile taxa. Among both groups, it's important to recognize that the most abundant members were like both low magnesium calcite and high magnesium calcite, low and high. Aragonite was not as common in the rocky intertidal as either of these two types of minerals. Among mobile taxa, the two most common low magnesium calcitic species were a lot of the limpets that are included in the Ladia complex, and then all of these small little grazing snails in the liverine complex as well. Among the high magnesium calcitic group, our mobile taxa include the purple urchin, Strongylus intronus purpuratus, and this cute little buggy looking critter, I can see it right here. And then we have on our sessile taxa, low magnesium calcitic species include the California mussel, Middleus californianus, barnacle, Nautilus glandula, both major space occupiers. And then on the high magnesium calcitic group, it's a little bit interesting in that we have algal representatives here as opposed to faunal representatives elsewhere. But the largest group of high magnesium calcifiers were the coralline algae, whether they be erect or foliose or crustose. How they line out in space? This gives you a bit of a sense. So up here we have density, which is the number of mobile organisms that were encountered during these surveys across all of the sites. This is percent cover, so this would be all of the sessile organisms that were encountered across the sites. And I've broken them down into the three main types of calcium carbonate. What you can see is that first, among the mobile species, by and large dominated by low magnesium calcitic species. And if we look at the sessile species, again, we have fairly high numbers of low magnesium calcitic species, but greater numbers as well of the high magnesium calcitic species spread throughout. And aragonite, a little representation towards the south, and a little bit scattered throughout up here, but most of the system is driven by low magnesium calcified calcifiers. The other thing we pick up is that there are definitely different patterns, right? That we don't have even distribution, a straight line all the way across, but we actually see more of this kind of shape action going on, which suggests that there may be areas that are favorable to calcifiers and areas that are less favorable to calcifiers. I'll summarize this chapter so we have time to cover some of the other details, but really we did see that calcifier distributions vary widely across the California current large marine ecosystem. We found that some sites represented hotspots of calcifier diversity or abundances, and these may serve as refugia for ocean acidification conditions. It's not to imply that we know what the OA conditions at those sites were like, but it suggests with high calcifier abundances that perhaps the water may be favorable to them. And then we also found those areas that with low abundances may indicate these high stress areas. So it's good information for stepping off and looking at other research questions as we go forward. And it also can be useful for targeting oceanographic studies that are focused on divergent environmental conditions by comparing these high abundance areas to these low abundance areas as well. So it starts to lay the map a bit for what's going on in our coastlines. So now let's talk about, rather than all species that we encounter on this coastline, let's get into the details of how one species responds in its natural environment. And in this case, it's the purple sea urchin. But as I mentioned, I looked at California mussels as well in a similar way. So before we dive directly into the urchin results, I want to compare and contrast mussels and urchins just a little bit. Both are similar in that they're calcifiers. They're major intertidal space occupiers. They're sessile primarily. I say primarily because urchins are typically recognized as being a mobile species, but in the intertidal, where wave conditions are harsh, they're more apt to hold on and stay in place in the rocks, and they feed in a different way that I'll mention in just a moment. And they both are important in integrating nutrients into intertidal food webs as well. But as we look at the details of some of those aspects, we can see that they are indeed very interesting to study, and they're divergent and interesting to study because of the divergence they have in those characteristics. First of all, they're very different phylogenies. So Echinodermata is the phylum for purple sea urchins, and mussels fall into the mollusk phylum. The primary type of calcium carbonate is also different between the two groups. So purple urchins have the high magnesium calcite form, the most soluble form, whereas California mussels have a double layer that is composed of aragonite and low magnesium calcite. Among calcified structures, urchins are interesting in that they have three different calcified structures. So I'll talk in a bit more detail about those three structures, but it differs from mussels in that mussels only have the paired shells as their calcified structure. If we talk about the occupied zones, you find urchins lower in the intertidal. California mussels make up dense beds in the middle of the intertidal. And in terms of the nutrients that they integrate, purple urchins are reliant upon macroalgae that drifts into the system, and they capture these blades of algae and then feed on those blades that are brought in and drift into the system. Whereas mussels are filter feeders and therefore more dependent on planktonic supply of food sources. So they integrate different forms of nutrients into the intertidal food webs. They do both modify the habitat. Purple sea urchins do it mechanically, where they actually use their spines and in some cases mouth parts to excavate burrows. And those burrows are thought to provide moisture during exposure at low tide, and they also help ameliorate some of the effects of wave action washing over the top. Whereas mussels don't actually alter the substrate, but by attaching to the substrate, create this incredible 3D structure that then acts as sort of condominiums for a whole variety of organisms that live deep inside the mussel beds, and are these infaunal populations that are dependent upon the structure provided by the byssal threads that attach these mussels to them. So we'll start out focusing on how sea urchins respond. Central question that leads to two follow-up questions. And the first question is, how do aspects of Strongylus and Trotus purpuratus performance vary in space and time? Do we see that there's variability that's really sizable enough to suggest the effects of environment? And if we do, how are those differences in their performance influenced by local pH conditions compared to temperature and food consumption? The approach that we took to this was studied over the 2012-2013 upwelling seasons, so again, roughly through October. And we used 4 rocky intertidal sites. These sites were chosen on coastal headlands and at capes to try to capture the range of oceanographic variability that has been well studied throughout the system. And at each of those sites, I had 4 replicate cages of urchins and 10 urchins in each cage. So you might be able to see the little purple urchins in their nice little cage. And these cages were mounted in tide pools where there were existing sea urchin aggregations. So you find in the intertidal, urchins tend to inhabit tide pool zones. Not all tide pools. In fact, at one of my sites at Strawberry Hill, they're only located in a single tide pool. But I worked within that tide pool because I knew that environment was hospitable for urchins. And then I went back and I fed these sea urchins roughly every 2 weeks during low tides throughout the course of the study season. Why feed them? Well, measuring drift algae that's washing into a system and is available as a food supply is notoriously difficult to do. So the best choice, the best alternative I had was to cage these guys and hand feed them. And what that involved was each tide series collecting roughly 250 grams of Saccharina sessilus, one of their preferred algal food sources. And then I would provide that to each cage, and I would collect what was left in each cage, and then I could measure the amount that I put in, how much I recovered 2 weeks later, and that gave me a sense of the urchin consumption. My sense is that a lot of that it was indeed consumption because the blades that I would collect that were left over did not show signs, very obvious signs of decay. In fact, they often had margins of herbivory around the algae. So it was clear that the urchins were indeed eating away on them. And then we also measured pH and temperature using 2 different types of pH sensors that are based on the same technology. One was the GFETs developed by Gernot Friedrich out of Macquarie as part of the Omegas program. And then some CFETs developed by Todd Marks out of Scripps at UCSD. And we have 1 logger per site. And each of those recorded pH and temperature over the course of the season. looked at a variety of responses for urchins. I was first, of course, very interested in calcified structures. As I pointed out, urchins have 3 different calcified structures. They have spines that are around the outside, and then holding those spines in place and holding the rest of the organs in place is the hard test on the inside, often thought of as the urchin shell. And within that test is the Aristotle's lantern, which is just this really coolly engineered structure that operates as their jaws inside. And so looking at these 3 differentially could be very interesting because the spines are obviously most exposed directly to the ocean waters around them, whereas the test is less so, and Aristotle's lantern even less so. So we may see differences among these 3 calcified structures. I was also interested in looking to see if there were changes in geometry, which can suggest the breakability of urchins as well. So I looked at changes in height or diameter to get a sense of kind of overall size of urchins from one location to another as well. And then to see if we had effects that fell outside of calcification, I looked at reproductive tissue in the form of a gonad index. The gonads here are these bright orange gonads. They're really highly prized for sushi in the form of uni. If you're eating uni, that's what you're eating. So I looked at the gonad index as the dry weight of gonads over the total weight of the organisms to see if perhaps there's some sort of energetic trade-off from calcification to softer tissue. So here's the results. This is the general layout for the next few slides. You can see that I've got a map here on the right-hand side, just to remind you where the sites lay. The sites are listed in each from north to south. I've got 2012 in the lighter gray, 2013 in the darker gray. And what jumps out immediately is that for all of the dry weight responses, you can see that 2013 outperformed 2012 generally. So there's definitely a year effect. And it turns out that the site near, a little bit of cutoff here on my slides, but the site near interaction was important for all of these responses except for the interaction on the lantern dry weight over here. In general, you can see that we have heavier organisms towards the north, where lantern dry weights are highest at Fogarty Creek and Strawberry Hill, as our test dry weights, spine dry weight, and that carries over into 2012 as well. Although we got some interesting performance out of Cape Blanco in 2012, where it was one of the higher performers in 2012, but not so in 2013. Looking at test height, we see similar patterns where we have higher performance in 2013 compared to 2012. And we also see that we have larger organisms towards the north generally, except for this exception of Cape Blanco in 2012. So the patterns that we saw for calcification weight carry over to the geometry of the test itself. And now looking at the gonad index, we can see that there's not very much variability with 2 major exceptions. Huge gonad production at Fogarty Creek and Cape Blanco in 2013. But other than that, relatively consistent from 2012 across the Strawberry Hill at Cape Mendocino in 2013. So what does this mean in terms of the influence of the environment? There's a lot on this slide right here, so I'm going to take a little time to explain it. We can talk about additive models that influence the outcomes of responses. We have a response that's dependent upon the factor of one item plus the factor of the second item plus the factor of the third item. And that's what we would call an additive model. It's explained by adding these 3 contributing factors together. And we can explore various additive models and look for which additive model best represents or best describes the responses that we see. Perhaps it's only 2 of the 3 items that best describe it, or even a single item that best describes all of these possible options. And so I ran a series of stepwise model selection using what's called the AIC method, and took a look at how important pH, temperature, and food consumption were as additive factors in explaining all of these responses. And it turns out that in all cases, all 3 factors were important. Okay? So that's the first thing I want you to realize is that there's no NAs over here. We have all arrows. So everything was actually, all 3 factors were important to each one of these responses. Now, how well those factors explain variation in those responses is measured by this R-squared value. So you can see that the inclusion of those 3 factors explains spine dry weight the best. That's about 0.58, where if it explained it perfectly, our perfect value would be 1. But this is about, explains about 58%. Whereas the gonad index is a much lower number that's explained. Right? So we've done a much better job of capturing variation using pH, temperature, and food for spine dry weight than for gonad index. Now the direction of these arrows. All over here. This is the direction of the influence of those factors as they correspond to increasing performance. Say that again. The direction of the influence of those factors as they respond to increasing performance. So, increasing spine dry weight is driven by decreasing pH. And that same trend holds through for pH for all of the factors. Higher test dry weights at sites with lower pH. Higher lantern weights, higher test height, higher test diameter, and higher gonad index at sites that have lower pH. Comparing that to temperature, we see the same pattern for temperature. Again, all arrows are pointing down. So we get increased performance of each one of these parameters as we are at sites with lower temperatures. And the opposite pattern holds true for food, where we get increased performance with an increase in food availability, which makes total sense, right? Feed things, they grow better. But what's really interesting are these effects of increased performance at decreased temperature and increased performance with decreasing pH. So those last models, we can step back for a moment, considered the best model fit. And the best model fit was the sum of each of these factors. We can run another method called hierarchical partitioning that takes a look at all possible additive models, those that include just a single factor, those that include pairs, and those that include all 3 factors, and get a sense of the overall contribution toward explaining variation from these factors. So here again, we're looking at the 3 aspects of spine dry weight, test dry weight, lantern dry weight, and you can tell the overwhelming influence in all cases is food. These guys fed well, they ate well, they grew well. Okay. What we can also note though is that pH, which is really the question that we're after, is what is the effect of pH on these organisms? It's comparable to the effects that we see from temperature. So pH is an important aspect to consider when we're looking at the performance virtues. Looking at the same thing for test dimensions, the same patterns hold true. Dominance by consumption of algae, pH and temperature roughly on the same scale. And then we look at the gonad index, where again, dominance by algae, some effect of temperature and pH as well. But again, I want to point out that it doesn't do much to explain the variation that we see in general. So, to summarize quickly, how do aspects of sea urchin performance vary over space and time? We see that across sites, they were generally heavier in 2013 compared to 2012 and larger. They were more robust towards the north than they were towards the southern sites. Calcified weights varied more than the aspects of test geometry. And gonad index was actually relatively consistent among sites and years. So what this really suggests is that pH is having a primary effect on the calcified components and not on other tissues inside of the organism. In terms of the influences of pH among other factors, we see that food is the dominant effect when I essentially provide them as much food as they want to eat. Because each time I returned to the cage, just about every time there was algae left over, so they had plenty of food throughout the course of the season. But we also see that pH and temperature have similar explanatory contributions, although lower than full food provisioning. Among responses, we also see that environmental factors explain the greatest variation in calcification weights. So we do see those effects on calcified components. And we see increased performance at sites with lower pH, which contradicts that prediction that we would expect for calcifiers in general, of reduced performance under pH stress. It's an interesting story we have coming out of the urchins. Now the question is, how does another important intertidal organism compare. So I've asked similar questions about the mussels as well, including how does the California mussel performance vary over space and time, and how is it influenced by local pH conditions when compared to temperature and food consumption. This study was done over a bit larger scale. This was part of the Omega's consortium approach. So I was fortunate to work with collaborators out at UC Davis who managed these sites, UC Santa Cruz who managed these sites in the intertidal, and UC Santa Barbara who managed the sites further to the south. It gave me a much larger latitudinal range for the study. It occurred during the 2011-2012 upwelling seasons. We looked at 8 rocky intertidal sites, again hoping to capture the range of oceanographic variability across all of those regions. And in this case, we had mussels that were put just directly onto the rocks, not in tide pools. We put them into the middle of existing mussel beds. We had 5 replicate plots of mussels per site. Right here, we had about 50 mussels in each replicate group. And then we transplanted the mussels for about 5 to 6 months during that upwelling season. A portion of this study, we also transplanted mussels out onto moorings as well to get a sense of the difference between intertidal and constant depth exposure on moorings, but I won't talk about that today, so feel free to ask questions if you have more about that. So with this setup in mind, we also had to monitor the environment, and here we used the GFET logger developed by Gernot Friedrich for the Omegas project with 1 logger per site. We measured temperature using these cool little epoxy robo mussels that involved a little temperature logger that was sunk into the epoxy, and the epoxy carried the thermal properties such that as waves splash over, it would essentially integrate and align with mussel body temperature as it's exposed to these different waves coming over the top. These were developed by Brian Comey at Northeastern University. And then we also looked at food availability in terms of chlorophyll A in the water column. And this is the concentration of chlorophyll A. And we didn't have consistent bottle sampling from one site to the next over each of these study years. So instead, we took climatological summaries over the course of several years of the bottle samples that were done at these sites, and then applied those climatological summaries to our analysis here. In terms of responses, I looked at seasonal growth, where we put a notch in the end of the shell, collected them at the end of the season, and measured how much shell grew beyond that notch, and then corrected it for initial length. The experiment of the study. We looked at condition index, which is a measure of tissue dry mass per total dry mass, and could be indicative of energy allocation to the meat part of the organism. And it could also be an indication of predator resource quality, again, meaty mussels for predators as opposed to more lightweight mussels. And then looked at shell thickness as a measure of shell dry mass per unit length. That could be indicative of predator vulnerability to crushing organisms or those that drill through the shells. Our results are set up similar to the urchin experiment in terms of the slide layout. You see the sites on the far right-hand side. Seasonal growth starting in the upper left-hand corner. There's quite a bit of variation. In fact, we have as much as about 1,300% of variation. When you look at these really low growers here at Lompoc Landing in Southern California, compared to these really strong growth here that we see at Bodega Marine Labs in Northern California. We also tended to see quite high growth towards the north at our sites up here in Oregon. Condition index, we see very high performance towards the north here as well, and not quite a suppressed performance at some of our southern sites compared to growth components. And shell thickness is, while still variable among sites and years, is more consistent across these than perhaps seasonal growth. Again, about 1,300% variation here. Here we see about 300% variation, and here maybe 100% variation overall. So, less variation in shell thickness. Back to our additive models. So again, looking at the importance of pH, temperature, and this time chlorophyll A as a measure of food availability. And in this case, temperature dropped out of the model, out of the most parsimonious model for shell thickness. What was left behind was just pH and chlorophyll having an influence. The direction of influence is interesting again, where we see the same trend of seasonal growth being higher at sites with lower pH. condition index being higher at sites with lower pH. Temperature, however, had the opposite effect. So unlike the urchins, we see that performance increased at higher temperatures. And we also see an increase with food availability as well. Looking at the environmental contributions now across all possible additive models, we can see food matters. Things that eat a lot can grow a lot. It's a good check. But we also see that pH is in fact an important contributor towards explaining the variation that we see, particularly when it comes to condition index and seasonal growth. And in fact, for condition index, it explains almost as much of the variation as chlorophyll A does, which is very interesting. So pH is indeed an important consideration in the effect on its organisms. So to summarize the muscle results, size and performance can vary up to as much as 1,300%, both among sites and between years. We did see the greatest variation in seasonal growth, followed by variation in condition index, and not as much variation in shell thickness. And then source population, which I'll mention briefly. We can talk about that offline as well. But in 1 year, we actually mixed source populations, among the sites to get a sense of prior exposure. So we took mussels from one location in Oregon and distributed them across all the sites, and got a sense of that prior exposure. And we did find that source population did mediate some responses, suggesting that there is some carryover effect from prior life history that can play a role in mussels. And how are the differences in performance influenced by pH among other stressors? We found that phytoplankton productivity had the greatest influence on responses, with pH ranking second over temperature for both seasonal growth and condition index. pH is an important consideration in our system. We also found that among responses, environmental factors explain the greatest variation in condition index. And this perhaps suggests that there's this energetic trade-off again between shell maintenance and the quality of the body that's inside. From an organismal perspective, what good is thin armor? If you're really meaty inside, what good is very thin armor? Maybe it's better off to have thicker armor and a little less meat on the inside. Not that it's a decision they're making per se, but it could be an energetic trade-off that's part of their physiological response to changes in the environment. And we saw increased performance at sites with lower pH, again, countering the predictions based on carbonate chemistry dynamics. So, some concluding thoughts to tie it all together. First of all, we did see similarities in responses between the two species. Food availability explained the greatest amount of variation among responses regardless of the two species. We also saw that for most responses, pH contributes at least as much or perhaps more toward explaining variation as temperature. So pH is an important consideration. And we saw that responses were generally enhanced at sites with lower pH. And this is perhaps one of the most interesting findings of all, that we see this enhanced performance at lower pH sites. Some major differences between the two studies were that mussels exhibited greater overall variation in sea urchins. Here, sea urchins may be more robust to environmental variation. And the sea urchins, pH explained the greatest variation in calcified weights. So it looks like pH is hitting those calcified components. Whereas in mussels, it looks like pH contributed the most to the variation in condition index. So these organisms may be dealing physiologically with these environmental stressors in very different fundamental ways. General take-home messages from these two experiments is that compared to mussels, sea urchins were generally more robust to the influences of pH. Mussels may respond to natural pH variation with energetic trade-offs between shell biomineralization and tissue development. Both species appear to thrive under reduced pH. Which really leads to this fourth point, that we have a highly complex environment in the California current large ray ecosystem due to the upwelling processes that are affecting temperature, productivity, nutrients, pH, salinity, mixed layer depth. And so it really underscores the effort, or underscores the need for continuing efforts to better characterize the pH variability that we see in this system and understand how it works with the other dynamics of this system, so that we can start to tease apart really how much these are playing important factors. Why does OA matter? Turns out that there's a lot of money that's at risk here. Total fisheries for the U.S. are about $4 million per year. And if we break that down into separate categories, we can look at just mollusks. And about 18% of U.S. fisheries, based on 2007 U.S. domestic vessel revenue, is derived from mollusks. So you can think if you suffer from OA, it's going to affect this aspect of fisheries. Same goes for crustaceans. A very large percentage, about 30% of the fisheries revenue comes from crustaceans and lobsters. And then we can think about food web connectivity, where 24% of species are calcifier predators. So if you see harm or threats to the prey for the species, we can see reductions in those species as well. And then we also have top-level predators, so we need to consider the whole food chains and the potential impacts of OA. They have very dramatic financial impacts on U.S. fisheries. And I'd also like to point out that OA is just one of many stressors affecting the marine environment. So here we have increased greenhouse gas concentrations, driving increased CO2, which decreases pH. But we have evidence for perhaps intensified upwelling, increased storm frequency, sea level rise, water temperature, and a variety of factors that need to change, highlighting all the more reason why we need to continue to do experiments in ecological settings to get a sense of how all these things interact together to influence organismal responses. Final take-home messages. Hopefully this is what I leave you guys with walking out of here, is that OA is a major threat to marine ecosystems, and it does have some far-reaching socioeconomic impacts. Marine species inhabit variable pH environments, though, and they may be acclimated or adapted to such variations now. So it's important to understand those that acclimatory or adaptive characteristics as we go forward in research. Natural pH variations can influence ecologically important aspects of calcifier performances, but these influences are highly integrated with other environmental aspects. So it's a complex world that we live in, but we do see that pH is an important factor in that complex world. And so I really highly want to recommend that future studies consider the aspects of pH alongside other stressors to better understand potential outcomes in our changing cities. So with that, I'm going to thank, I can't possibly thank enough people really, but I'll try. Bruce always works a picture of you into that. Is that you? It is. Yeah, probably. Into one of your slideshows, so I thought I'd work it into mine too. I can't thank Bruce enough for his leadership, his quiet, calm demeanor that has helped me get through some very stressful experiences, and just his general approach to science has been really foundational to who I am as a professional today. I'd like to thank my PhD committee members. I do not have currently an 8-member committee. It's really not that big. I've had some changes along the way. I'm very blessed now to have Jane, Tarek, Kirsten, and Ricardo as current committee members, but I received back support from Sally, Hal, and Eric earlier on in my process. And I'm just so appreciative for the efforts that they've all contributed. Tarek has been a huge supporter in terms of my development as a statistician, so I'd like to especially thank him for that development. And Kirsten and I also had the honor of being office mates for 4 years, and so she really helped my development as a graduate student, and I wish you just terrific friendship as an office mate. Of course, all of the amazing grad students, past and present, I hopefully captured everybody. It's a long list, and it's an illustrious list that I'm very proud to be a part of, and a very fun group to hang out with here. We're a little nutty at times, but generally very hardworking and just incredibly knowledgeable, thoughtful group of people. So thank you all for your support throughout this experience. Huge host of Peace Corps lab members. We have quite a team upstairs, as I'm sure many of you know. But we really have kind of the mothers who keep us in line with Cindy and Kathleen. We've got the older brother who's going to beat us on the head if we do things a little off kilter, and that's Jared. Kristen does incredible behind-the-scenes work, really keeping publications going and keeping things collected and organized in the Peace Corps world. Annalise is a current postdoc that we have, who's contributing greatly, and then the work from current and past lab technicians, volunteers, etc., who've done tremendous amounts to support my work both directly and indirectly through community centers. huge host of undergraduate assistants. Graduate work does not happen without the help of undergraduate assistants, and I can't, I've thanked them all many times, but I can't thank them enough for their contributions to helping with collecting data. I'd also like to thank all my Omega's collaborators for the incredible work that's been done over that consortium's the last few years of that consortium. Many zoology friends who have since moved on from the department, but are all contributed greatly to their friendship and intellectual expansion of my skills as a scientist. The Integrative Biology Support Network, so the chairs of the department since I've been here, started out with Bruce and Joe, a little bit with Ruben, but mostly Bruce and Joe, and then Virginia Weiss has taken up the mantle and carried it very strongly forward. Of course, we're supported by wonderful folks in the office with the 4 T's, and now Tori's been dropped out. I can spell her name wrong. She's hasn't been gone that long, I spelled her name wrong. But she's been replaced wonderfully by Jane, and it's just a wonderful team of women to work with who really help keep the wheels in motion for the department on many facets. And I also thank Bob Mason, Devin Quick, and Lauren Kings for the support that they do through various aspects as well. And then, of course, there's lots of money that goes into research, and I've been fortunate to be funded through a variety of organizations, both through fellowships and grants that I've received, and then funding through Bruce's Lab as well. Of course, I couldn't have done any of this without the support of family, of many who are sitting in that middle row right there. My mom and my sister, my grandma and grandpa, and my little brother, and wish my dad was sitting here, but he's been a tremendous influence throughout all of this for me, and it's really a major reason why I've been able to keep pushing forward through all of this stuff. And of course, I couldn't possibly have done any of this without the love and support of my immediate family, my wonderful wife Heidi, and our beautiful daughter Sierra. Sierra has been familiar with the Rocky Inner title since she was a wee little one, and has made many trips out to the beach with us. In fact, was helping me measure muscles when she was, well, only a couple weeks old at that point. So she's already well on her way to becoming her own marine biologist someday, and we're working on that. But thank you. And lastly, I would like to thank all of you for attending. Thank you for being here.
We'll go over, but let's just have a few questions. The committee members should have their questions afterward. Any questions?
So, with the importance of food and everything, how much do we know about how much pH might affect primary protein?
So there's been a huge number of studies that are also looking at planktonic organisms as well. And phytoplanktonic organisms are, one theory is that the increase in carbon dioxide may actually be beneficial for organisms that utilize carbon dioxide for primary production. But it's been found that many of those organisms already have what are called carbon concentrating mechanisms in their physiology that allows them to bring in enough carbon for growth and to sustain growth. And so the addition of additional carbon dioxide does not really do much to bolster the productivity of those organisms. There are other reasons to suspect climatic changes will affect the productivity, but it looks like ocean acidification is not as strong an effect on primary producers as it is on calcifiers and other organisms. Yeah, Nate.
At the beginning of your talk, you had mentioned the larvae are really getting hit hard by this, and your data were quite stark in the opposite direction. I was wondering if you had...
Yeah, so with the Omega's work, there's been work by collaborators on the larvae of both the purple urchins and the California mussel, and show that pH is incredibly stressful on growth, on survival, on several different aspects. On shell strength is another aspect where low pH truly has very detrimental effects. Some other studies that have looked at juveniles suggest that these effects are suppressed, that there are still negative impacts on juveniles, but not to the degree that there are on the larvae. I'm talking juvenile purple urchins and California mussels. My results are on adults. So perhaps there's some sort of filter that's going on through life history where those that are susceptible are then essentially removed from the population, those that make it through the filter are then well adapted and can accommodate these swings in pH variation. Yeah, Alec.
So, there's been a lot of recent health reports showing detrimental effects of acidified water, low pH water on adult calcifiers. So what do you think is the disconnect between the reports showing that calcifiers are definitely going to be impacted by OA and the stuff that you've done? What's the difference and how are they linked together?
I think it really comes down to the complexity of the environment along the California current system, where these changes in pH, temperature, upwelling, food availability are all fairly tightly correlated. So teasing apart the direct effects of each of those is a statistically challenging job. The approach that I've taken has been a fairly broad brush approach to get a first sense at do we see pH as being an influence. But I think you're right, that there's definitely more work that needs to be done in how we tease apart those components in a natural environment so that we can get more at the direct effects in the natural environment. And perhaps then we will see that these performances that are related to sites with lower pH are truly driven by other environmental characteristics that make them more robust to those pH effects. Whereas if they suffered those low pH effects alone, they may be detrimental effects.
Any others? Okay, well, thank Jeremy and